So this was the last part we had studied about histones and non-histones protein. Okay, histones. How many histone molecules come together to form that structure called nucleosome? How many histones were there? You only had answered.
How many?
How many histones proteins were there together to form that?
That is before.
Yeah. How many were there total?
1, 2, 3,
4.
4.
4 into 2, 8. That's why it was called as octamer.
I just do HP.
Now, okay, then this was already done about histones. So this is the 8 protein. So like this, they are joined. Many nucleosomes will be joined together. Nucleosome will join together to form chromatin fiber. Chromatin fiber will come close to each other to form chromosome. Now these chromosomes are visible during your cell division. During prophase, metaphase, anaphase and telophase. Okay, these chromosomes are visible. So let's come to the next point. This is done non-histone protein. Euchromatin means which is transcriptionally active. Heterochromatin is not active. Means from euchromatin you will get a protein. Means it is a long DNA stretch. Some part of DNA will act as euchromatin. Some part will be heterochromatin. The part which acts as euchromatin will give you protein after the process of transcription translation. Heterochromatin will not give you protein. Okay. The properties of genetic material, DNA versus RNA. Who solved the debate question whether protein or DNA is the genetic material? Hershey's and Chase. They did the experiment on protein and DNA. So they found that DNA was the genetic material.
Chase is a girl.
Yeah. Hershey and Chase. Okay, this was the experiment. They labeled the protein with radioactive sulfur, and at the end they didn't find any radioactivity. They labeled the DNA with radioactive phosphorus. This was injected in the cell. The new phage you got will be having phosphorus, and there was radioactivity.
So this video you shared.
Yeah. Okay. Next is in some virus, RNA is the genetic material. Although DNA was found to be genetic material, there are some viruses where RNA is the genetic material. Example is TMV, tobacco mosaic virus.
You did this in 11.
Yeah.
That's what I was thinking.
And the other one is QB bacteriophage. So you can see here RNA is the genetic material. This is RNA. Here RNA is the genetic material. You see, this is the what is the difference between RNA and DNA? Yeah.
The thymine
Thymine.
and
Uracil. First is thymine is replaced by uracil in RNA. Which sugar is present in RNA? Ribose. Which sugar is present in DNA? Deoxyribose. So these are some differences. That is, if you see here, it is cytosine, guanine, adenine, and uracil. But here it is cytosine, guanine, adenine, and thymine. Okay. The other thing is that OH is extra in RNA. That OH will be extra. That's why it is DNA is having one O less. That's why it is called as deoxyribonucleic acid. Deoxyribose nucleic acid. The next, what is there? Okay. Then a molecule that can act as a genetic material must fulfill the following criteria. What are the properties of the genetic material? It should be able to generate its replica. It should be able to replicate. It should be able to make its own new copies. Second, should be stable chemically and structurally. It should be stable chemically as well as structurally. It should not change its structure or chemical composition. It should provide the scope for slow changes. There should be some slow changes. These changes are called as mutation that is required for evolution and should be able to express itself in the form of Mendelian characters. It should be able to express itself in the form of Mendelian characters. Means from father it should go to the son or daughter and should be able to express. Next, DNA and RNA fulfill this requirement, but protein fails. All these requirements which are given here are fulfilled by DNA and RNA. That's why DNA and RNA is the genetic material. Here. DNA to RNA and protein. This is the central document. So DNA, what happens? This will unwind. When there is replication, this will unwind and form new copies. This RNA also can make copies of itself, but protein, it does not have anything to make copies. So DNA and RNA fulfill the requirement of genetic material. Okay. Next is genetic material should be stable enough. Very important. It should be stable. Change, which should not change with change in stages of life cycle. For example, from metaphase, prophase, anaphase, telophase, it should not change during that time. Whatever genetic material is in one cell should be copied exactly in the other cell. If anything misses, then there will be mutation and the characters will also differ. So also it should not change with age or change in physiology of the organism. Okay. What was noticed in Griffith experiment? This was Griffith experiment.
Yes.
He took live aspirin, mice was alive. He took live aspirin, mice died. He used heat kill aspirin, mice is alive. But when this heat killed aspirin were mixed with aspirin and then injected, the mice died. So what must have been what is kept safe in this heat killed aspirin?
DNA.
DNA was stable. It was stable. Even the heat was not able to destroy it. Okay, even the heat was not able to destroy it. This DNA was alive in this. Okay, DNA was stable there. That genetic material got transformed in R strain, and you got this mice kid. So DNA is stable. So what this is now can be easily explained in the light of DNA that the two strands being complementary. So what must have happened? DNA is having two strands. So they must have separated. When heat is supplied, the two strands, there is the hydrogen bond. When we supply heat to a bond, the bond breaks and they separate. But what happened here, when the favorable condition arrived, the two strands joined again and formed the DNA molecule back. That's why this DNA molecule was found in this mice blood sample. If separated on heating, came together when appropriate conditions were provided. So what is extra in RNA and DNA? What is extra in RNA than in DNA?
H.
O. In ribose, there is OH. In deoxyribose, there is no O. Okay. On which carbon it is? Second carbon. Second carbon. See that O is on second carbon. Okay. 2 OH group. Then it's a reactive group. Okay. So this 2 OH group, no, it is extra. No, anything extra means it will react. Means it will have extra energy. Extra energy means it is reactive. Anything which is not having any energy, it will not act. It will sit quietly. No reaction will be shown. Okay. We have studied in periodic table the reactive metal and non-reactive metal. The reactive metal will have extra energy. They have extra electron which they want to give out. They don't want that extra electron. They want to either give out. So that's why they react. For example, sodium, potassium, they all have extra. So is real. RNA is also known as catalytic. RNA also is a catalyst. Okay. They are called as RNAs. Enzymes are called as cellulase, lactase, pectinase, polymerase. So fructase, lipase, protease, DNase. So like that there is RNAs. Okay. Therefore, DNA chemically is less reactive and structurally more stable than compared to RNA. So if DNA is less reactive, that means it will be more stable. It is less reactive. So, compared to DNA and RNA, so which is better? DNA will be a better genetic matter. So DNA is more stable. DNA wins over RNA. Okay. We'll stop here.